Hello everyone. So continuing with the cognosy subject, we are already started the topic in general pharmacognosy, chemical nature of crude drugs. Now in that, after alkalides and glycosides, next one important is volatile oils. Now most of the volatile oil containing drugs also we have covered, and you might be remembering about the carminative drugs, for example coriander, fennel. So what are volatile oils that we will see in first few slides. We will try to understand the definition and some important aspects of volatile oil. Now the odorous principles, volatile principles of plant and animal source are called as volatile oils. They are liquid in nature, they evaporate when they are exposed to ordinary temperature, and therefore they are also called ethereal oils. The another name for these oils is essential oils, as they are acting as essence, or we can say flavor. So they can give a very good and remarkable flavor, isn't it? So they are essences or concentrated constituents of the plants. So again about the volatile oils, that these are essential oils, and as they are called volatile oils, so they are volatile in nature. They get easily evaporated. They differ totally from fixed oils in chemical properties, in physical properties, isn't it? They are secreted in oil cells, in secretion ducts or cavities, or in glandular hairs. So in depending upon the plant, they are present in different cell structures, isn't it? So that also I will let you know. They are frequently associated with other substances such as gums and resins, and themselves they tend to resinify on exposure to air. Now these volatile oils, they are volatile in nature. So same thing, that they are totally different from fixed oils. They are getting easily evaporated when they are exposed to air, whereas fixed oils like coconut oil, RHS oil, groundnut oil, they don't evaporate. They are fixed oils. Then they are present in different cell structures. For example, oil cells, cinnamon bark, dalchini, isn't it? So in oil cells, in parenchyma cells, the oil is present. Then oil glands, which are present in clove. We have seen the transverse section of clove flower, isn't it? Then secretion ducts, for example, vitae cells. So in transverse section of coriander fruit, we have seen that there are two vitae cells, isn't it? So this vitae cell is the main character which contains the chemical constituent of the drug. Then glandular hairs also are there in some of the drugs where the oil is present. For example, chamomile, or we can take another example of black pepper also, kali miri, isn't it? So glandular hairs contain main constituent of the drug. Then they are frequently associated with other substances such as gums. So some oleogum resins are there, isn't it? uh, in combination with the gum and resin, they can be present in most of the drugs. Now we must also know the what is the composition of volatile oils. So chemically they are mixtures of hydrocarbons and oxygenated compounds. Oxygenated compounds which are derived from hydrocarbons. Now the odor and taste of volatile oil is determined by these oxygenated constituents which are soluble in water, but comparatively these are more soluble in alcohol. We'll see some important physical properties of volatile oil. Now, they differ greatly in chemical constitution, isn't it? They have got strong characteristic odor, they have high refractive index, most of them are optically active, their density is generally lower than that of water. Then volatile oils are immiscible with water, but they are sufficiently soluble to impart their odor to water, isn't it? So, although they are oils, but they are not miscible, but still they can, being they are having little bit solubility, so they give their own odor to water also. Now, aromatic waters are dependent on this slight solubility. So, aromatic waters, the preparation that you have already prepared in pharmaceutics practicals, isn't it? So, these aromatic waters they contain volatile oils. Now, these volatile oils they are liquid, they have pleasant smell, as we have seen, they have strong odor, they are steam distilled, they are colorless liquids, isn't it? One of the exception is chamomile oil, which is violet in color. Then they are mostly lower than that of water, isn't it? But some, for example, clove, cinnamon, they are heavier than water. They are immiscible with water, but they have got little bit solubility and they can impart their own fragrance, pleasant smell to water. They are soluble in common organic solvents and lipids also. They have high refractive index and most of them rotate plane polarized light. They may darken in color when exposed to light. So due to oxidation and resinification, there are chances that their color, original color may get changed and they may become dark. Now, classification of volatile oils, isn't it? So, according to the important functional group which is present, based on that, they are classified. One is hydrocarbon volatile oil, where the example is turpentine, isn't it? And another example also we can give of black pepper. Then alcoholic volatile oil, or we can say alcohol volatile oil, where the example is peppermint, cardamom, coriander. Then aldehyde volatile oil, for example cinnamon, then bitter orange, isn't it? Orange peel, lemon peel. Then phenolic volatile oil, for example clove, then thymine. Then ketone volatile oil, example is camphor, spearmint, caraway. Then phenolic ether volatile oil, where we can give example of anise oil, then fennel, then myristica, that is the nutmeg, zephyr, then oxidized volatile oil, for example eucalyptus, chenopodium, then ester volatile oil, for example rosemary, and miscellaneous will include anethum and allium. So anethum, that is the fennel we can call, and allium, that is the garlic. Now we'll see the method of isolation or extraction, how volatile oil can be extracted, and there are different methods depending upon the type of the drug, isn't it? So this depends where oil lies, isn't it? So as I said that it is present in various cell structures we have seen in the properties, isn't it? Different cell structures are there. So based upon the plant part and cell structure, the method can be variable. Then steam distillation can be followed for extraction, isn't it? Then one can go for hydro distillation also, that is water distillation, isn't it? Or it can be obtained, the oil can be separated by cold expression also, by applying pressure to the citrus fruits, isn't it? Or whatever the rasping process is there, then they can be obtained by pouring cold water over and then separating oil and water, isn't it? Now, there are three techniques of obtaining or separating oil from the drug. One is distillation. Now, in this distillation, it may be water distillation, or it may be water and steam distillation, or direct steam distillation, isn't it? So, in distillation also, there are different modes, isn't it? So you can see the uh, distillation of essential oils, so how it is carried out, and distillation plant is needed, distillation unit is needed, isn't it? So in actual practice, there are three different modes, isn't it? Depending upon the condition of the plant. So water distillation, which will applicable to such plants where uh, they are dried initially in the air, and the constituents, they not get decomposed or degraded by boiling up to 100 degree centigrade. For example, turpentine oil. Then water and steam distillation for such plant material which undergoes degradation by direct boiling, isn't it? And therefore steam is used along with the water. For example, clove oil and cinnamon oil. So they are to be initially first macerated with water for several hours before steam distillation. Then direct steam distillation, it is applicable to fresh drugs, isn't it? That is loaded with sufficient natural moisture, and there no maceration is needed. For example, peppermint oil, spearmint oil. The freshly cut drug is added directly into the distillation flask prior to steam distillation. So, so this is the distillation unit you can see, isn't it? Now, next method is enfleurage method. There are three techniques, isn't it? One is distillation, other is enfleurage method, and third is equili method. All three we are going to discuss, isn't it? Now, enfleurage method, this is mostly used for the delicate flowers. So flower plants, isn't it? So flower parts which contain oil, isn't it? For those, this method is used for isolation of the drug. Now, the flower petals are used, isn't it? And they are mechanically spread on the layer of fatty material, isn't it? So you can see how the wooden frames are there, glass plates are there, isn't it? And on these glass plates there is a fatty layer which is deposited, and this fatty layer is inert one, isn't it? So in this infrared method, the perfumes which are used, isn't it? Mostly those perfumes are obtained by this enfleurage method, isn't it? So the glass plate is covered with a thin coating of inert fat, odorless fat. The freshly cut flowers are individually laid on fat, which in time becomes saturated with their essential oils, isn't it? So in this slide you can see this is the oldest method which is used for extracting essential oils. It involves placing the flower petals on layer of glass that is first spread with thin layer of fat called chassis, isn't it? So inert fat is to be used, and on that fat layer the flower petals all are deposited. And then what happens? This fatty layer gets saturated with the volatile principles that are present in flower petals. This volatile oil diffuses into the fat, and then the fat is collected and oil is extracted from the fat using alcohol. So this is the influence method which is used for most of the drugs which are flower parts, isn't it? Now the third method is equally method, isn't it? Now this equally method, it is used to obtain citrus oils, isn't it? So extraction of citrus oils is done by this equally method. Now the oil cells in ring are ruptured mechanically using pointed projections, isn't it? By twisting raw material over them in clockwise direction. So you can see this, the fruits, say for example, as I said, citrus fruits, lemon oil, orange oil, isn't it? So whatever the peels are there, outer coverings, so they are rolled over the line with sharp projections. Now these sharp projections, they penetrate the epidermis and puncture the oil glands located in the outer portion of the peel. isn't it? The fruit is then pressed to remove oil from the glands. It is then sprayed with water that washes oil from the mashed peel, and resulting oil emulsion is separated by centrifugation, isn't it? So this is the third technique, that is called Equili method, isn't it? Now, important test for volatile oil. So general tests are that the section of the drug, alcoholic solution is added of Sudan 3 reagent. Observation is red color obtained by globules, which confirms presence of volatile oil. Then other test is thin section of the drug. A drop of alkaline tincture is added. Again it gives red color, which confirms presence of volatile oil. The next test is Sarkovsky reaction, where 2 ml of uh, drug, and to this 2 ml of chloroform is added and 2 ml sulfuric acid is added. Now what happens is chloroform layer appears red and acid layer shows greenish fluorescence, which confirms presence of volatile oil. Then next test is Lieberman reaction, where the 3 ml of extract is taken with acetic anhydride equal volume. It is heated and cooled. Then few drops of sulfuric acid are added, which gives blue color. It confirms presence of volatile oil. So these are some of the important tests of volatile oil. Then uses. So most of these volatile oil containing drugs, they have got important role in most of the diseases, in uh, treating most of the diseases, in industries, pharmaceutical uses, hasn't it? So one is therapeutically. In therapeutics, it has got important role. For example, oil of eucalyptus. Then it is a very good flavor, isn't it? As a flavoring agent, these drugs can be used. This is a pharmaceutical importance. Then in perfumery, where rose oil is used, then most of these oils can be used as starting material to synthesize other compounds. Then they can be used as antiseptic due to high phenols, also as preservative because these oils interfere with bacterial respiration. They can act as a antispasmodic, for example, ginger, lemon balm, rosemary, peppermint, chamomile, fennel, caraway, isn't it? They can be used in aromatherapy. So these are some of the importance, you can say, the volatile oils. They have got importance in medicinal uses. They have got pharmaceutical uses, and in cosmetics also they have got play important role. Now, functions of volatile oil. So in plants also they have got important role, like they protect plants from harmful effects of animals and insects, sometimes help in pollination through insects. Then other importance is therapeutic function. So as we have discussed in the previous slide, that in they have got medicinal uses also, like they can be used as a carminative. Most of the carminative drugs we have seen, they have got antispasmodic effect also, isn't it? They have got antibacterial role. They are stimulants. All volatile oil containing drugs, they have got stimulant property. They are anthelmintic, then expectorant. They are most of the antiseptics. They have got diuretic role, antipruritic, for example camphor, and in pharmaceutical field they are used as the important flavors and preservatives. So here we finish up volatile oils. Regarding all these important chemical groups, we must know the definition, classification, their occurrence and distribution in nature, then important identification test, method of isolation and extraction, and important uses. So if you don't understand, you can listen this video twice, thrice. Okay. I hope you will find this video lecture beneficial. Complete your notes also regularly. Thank you.